CRISPR is a revolutionary gene editing tool. It can be used to target DNA and change its very sequence, meaning it can alter the code which makes up everything about an organism, from how it looks to how it functions. It can activate or disable genes, DNA encodes, insert or remove a gene, and all of its power came from the awesome bacteria, E. coli. The CRISPR system was first encountered in 1987 when scientists came across something unexpected in the DNA or genome of the bacteria. They saw that there were unexpected sequences repeating and spaced at regular intervals, but couldn't figure out what these strange findings meant. Over the years, these sequences popped up more and more in different labs studying various single-celled organisms across the world and eventually were given the name CRISPR in 2002. This acronym described the structure of the mysterious sequences as clustered, regularly interspaced, palindromic repeats. Basically, this meant that the repetitive sequences were found bunched together in DNA, evenly spaced, and were palindromes, meaning they were read the same forwards as backwards. It wasn't until years later that scientists realized these spaces between the repeats matched virus DNA and was making the bacteria immune to that virus. Similar to how our bodies use exposure to disease to build up immunity to those infectious agents, bacteria used CRISPR as its own adaptive immune system to protect against viruses. Essentially, the microbe didn't want to continue being taken over and killed by its virus arch-nemesis, the bacteriophage, and so it learned to adapt. It used some helpful proteins with DNA cutting ability, called Cas proteins, or CRISPR-associated proteins, to take pieces of viral DNA, chop them up, and integrate them into their own genome, as the spacers within CRISPR. Bacteria would collect many of these protospacers from different viruses to make into their own CRISPR spacers and form an inventory of virus immunity. Equipped with their self-defense weapon, they would then express these CRISPR sequences and Cas proteins through transcription, where DNA is copied to form its single-stranded counterpart, RNA. The Cas RNA then went on to form Cas proteins through translation. And together, the Cas protein and CRISPR-guide RNA would target infectious viral DNA, disabling it before the virus could use it to multiply and kill the bacteria, while also allowing for more CRISPR spaces to be created. From these humble, but still really astounding beginnings, emerged the gene editing master tool we know now. To date, CRISPR-Cas has been used to edit the genome of various different organisms, from E. coli itself, to plants, to animals, to humans. Researchers have accomplished amazing feats, such as efficiently making bacteria produce biofuels, which could potentially replace fossil fuels like oil, engineering fruits and vegetables to last longer, and even making chickens lay eggs with cancer medication in them. CRISPR met the human body for the first time within the last few years, and it was a distant introduction at that. Cells were taken from the body, edited of their specific dysfunction with CRISPR, and put back into the body with hopes of curing sickle cell disease and cancer. In 2020, the CRISPR-Cas system was even injected into the eye of a patient with inherited blindness to try and restore sight, marking the first time CRISPR was put directly into the body of a human. However, the impressive speed with which the gene editing system rocketed itself into notoriety is paralleled with the massive safety concerns it raises, especially when considering its application in humans. When it comes to gene engineering, there is already a fair amount of skepticism about how safe the technology is. A good example is the fear around GMO, genetically modified organism, foods, which are modified by a gene editing technique with the intention of improving the product in some way. Some people believe GMO foods are harmful and can even cause cancer, but research has not supported that claim. Even so, many people strive to live their lives GMO-free, creating a massive market for the relatively unmanipulated foods. Now, consider the same perspective and worry applied to changing the DNA of humans. It's scary, but the reward is potentially so much greater. Gene engineering can potentially allow us to cure up to 7,000 diseases, each by targeting their one malfunctioning gene, or to manipulate one of the 40,000 proteins which determine our function and fitness. All of this is at our fingertips. However, the risk of editing the genome of humans is also greater. Our biology is much more complicated than that of E. coli or plants. Since the CRISPR-Cas system enacts these genomic changes, its use needs to be precise and highly regulated, or else it can cause damaging mutations. The CRISPR-guide RNA guide the Cas protein to DNA sequences which complement the RNA sequence, giving CRISPR relative precision in its editing. However, there remains a major concern: off-target edits. These can be caused by similarity in the target sequence to other, unintended DNA sequences in the genome. If the unintended sequence is acceptably similar to the intended sequence, the CRISPR can then enact its changes at that close, but not quite, sequence. This is worsened by CRISPR-Cas remaining in the host for too long, giving the complex more time to bind to unintentional locations in the genome. While these effects are minimized wherever possible, their presence still raises an alarm. Genes are highly interconnected, forming regulatory networks where the expression of a gene can affect the expression of a number of other genes within its network, kind of like a domino effect. Therefore, off-target effects can influence more than just the gene sequence they directly mutate. They can cause widespread mutation. Additionally, when using CRISPR for therapeutic application, it often needs to be restricted to the cell or tissue where the repair is needed. Targeting by the guide RNA is only specific to the gene sequence in DNA. However, different cells and tissues share the same, or mostly the same, DNA. This means targeting only a specific gene sequence could mean editing that gene in many different places in the body. This is definitely not ideal, as a genetic alteration can be beneficial in one cell and harmful in another. To prevent this from happening, there are some intervention methods to spatially contain CRISPR-Cas. Promoters are DNA sequences which enable the transcription of the DNA following them. A cell or tissue-specific promoter can be used to limit the expression of protein-coding genes to that cell or tissue. Therefore, this method can be used to control where the Cas protein is active. Additionally, the delivery method for CRISPR-Cas can be made location-specific. DNA encoding the CRISPR-Cas system are often packaged into disabled viruses and then directly injected into the site of interest. Different viruses are used for different applications, with each having a preference for a certain tissue or cell, or a variety of tissues and cells. However, the system is not always entirely spatially regulated, due to lack of strict, complete specificity of the promoter or of the delivery system. Expression of CRISPR-Cas can leak into non-targeted tissues, leading to unwanted edits. Further, a major cause of upheaval in the CRISPR field has been the prospect of germline gene editing. The germline is composed of the reproductive cells, those cells which pass on genetic information to offspring. Therefore, this kind of gene editing results in genetic changes which are then passed on to the next generation and generations following. This not only raises ethical dilemmas, it also presents serious safety concerns. As stated at the beginning of the video, CRISPR has not been applied and tested in humans for very long, and the results of these preliminary trials are still coming in. Therefore, we have no way to be completely certain of the long-term effects our manipulations will have on the people we test this therapeutic on, and introducing a harmful edit into the germline, intentionally or unintentionally due to poor spatial control, would result in future generations being left to deal with that mutation, no matter the effects. Finally, it is possible that our body, when exposed to CRISPR, would simply reject the system. Since our immune system serves to protect us from any foreign invaders who wish us harm, and Cas proteins are derived from bacteria, they could be perceived as a potential threat and disabled. Additionally, if the immune system recognizes Cas proteins as harmful, it could signal the body that it is under attack and create inflammatory conditions, essentially the body's attempt to make an environment inhospitable to invaders. This can cause damage to surrounding cells and tissues, in addition to rendering the CRISPR system useless. Regardless, candidates for this type of gene editing are extensively screened to account for all safety considerations, so if an immunity like this existed in a person, they would not be administered the CRISPR treatment and would need to seek an alternative therapy. In the end, this is still a relatively new field, which is rapidly growing and changing. While we do not know everything about CRISPR and its effects just yet, we are already making strides towards using it to advance human health while continuing to make the technology even better. Different variants of the CRISPR system have been created which work to get around the hurdles of CRISPRs before. These engineered spin-offs can improve efficiency and reduce off-target effects, while others can target DNA without cutting it, or even target RNA instead of DNA. This last development is very exciting, as it actually eliminates the genome manipulation concerns raised by DNA-targeting CRISPR systems simply by using a different Cas protein. Of course, the regulation of those transcripts and their expression still plays a role in biological function, but this is a less permanent form of manipulation that holds much promise in terms of medical therapeutics and diagnostics. Overall, the future of CRISPR is uncertain, but very bright. One day we might be able to eliminate diseases entirely, but for now, we will hope for steady advancement and positive results from the clinical trials in progress and to come.